So our next speaker is going to be Lester Rodriguez. And he is going to tell us about how we can use MRIs to image our brain.
All right. Hi, everybody. Can you hear me all right? Okay. So my name is Lester, and I'm going to be talking about neuroimaging, specifically a way in which scientists can look at brain activity without being invasive. So first, a bit about myself. I studied molecular and cellular biology at the University of Puerto Rico in San Juan. On the left side, you can see the campus. It's all green and sunny and nice, as opposed to the cold that we get here. And on the right side, you can see my lab. While I was there, I worked with animals. I worked with rats. You can see 2 rats in the picture there. And I studied anxiety-like behaviors in the animals. At some point during my undergrad, I decided that I wanted to get clinical research experience. And so I did an internship at the University of Pennsylvania's Center for Studies on Addiction. And that's where I first got introduced to human research and to neuroimaging specifically. I liked it so much that I thought I want to get more experience. So after I graduated, I did a postbaccalaureate fellowship. That's when you get your bachelor's degree, but then you're not sure what you want to do. So you do an extra year or 2 of full-time research, and you get paid to do it. So it's great to get more experience and to just figure out what you want to study after. And so now I'm a PhD student in Yale's Interdepartmental Neuroscience program, and I'm also a graduate fellow of the New Wusai Institute here for Neuroscience at Yale. So let's get started. Can anybody tell me some things that the brain is important for? Some examples. I'll take like 3 examples. Over here. So basically everything, so that's a good way to put it. Over here. Moving, thinking, yes?
Raising your hands to answer the question.
Raising your hands to answer the question. I like that one. That is 100% true. So, right, so your brain is important for a bunch of things. So movement, right? When you're running, your motor coordination, your sense of smell, fine motor coordination needed to write, storing your memories, processing your emotions, when you're talking and you're listening to each other and being able to interpret what people are saying. When you're doing sports, the coordination required to do sports, very complicated things like math. Your brain is also important for regulating bodily processes like breathing, heart rate, etc., your digestion, and also things like planning, right? Very complicated things that only humans can do because we have distinct brains. So to understand the brain, we first have to understand the body first. So your bodies are made up of tissues, and the tissues are made up of what we call cells. And so the key thing about these cells is that every cell has a specific form that facilitates a specific function in a specific localization. So if we're talking about the blood, we have blood cells that carry oxygen. If we're talking about the intestine, we have intestinal cells that help break up food. If we're talking about muscles, we have muscle cells that help you move and lift things, etc. Liver, we have liver cells, and in the brain, we have what are called nerve cells. Okay? So, like I said, in the brain, we have nerve cells, and the key nerve cell here is the neuron. And so neurons have this weird shape here, and the important thing that you need to know about neurons is they can communicate with each other with electrical signals. So if you were to visualize that, it will look something like this, right? So you have all these neurons in the brain, and you have the electrical signals jumping from one neuron to the other, and so they can communicate with each other, and they can be involved in all these processes that you guys talked about. So this is very complicated. We're talking about 86 billion neurons that are communicating with each other, right? So this is a lot of neurons, a lot of interactions, and we need it to be that complicated if we want to be able to do all the things that you talked about, that the brain does. So now that I just said, right, that the brain communicates, or the neurons communicate with electrical signals, can anybody think of a way in which we could measure brain activity? Somebody. Over here? Measuring the electrical signal. That's exactly right. We could totally do that. So scientists do that all the time. They use electrodes to look at brain activity. The problem with doing that in humans is we can't just put electrodes in your brain. So scientists had to come up with a way to look at brain activity across the brain without being invasive. And that's what I'm going to talk about today. Now, to understand this technique, you first have to understand that the brain has needs. the brain needs two things, nutrients and oxygen, right? So in order for it to function, in order for your neurons to function, in order for the cells to function, they need nutrients and they need oxygen. For this technique, this need of oxygen is key, okay? The question then becomes, how does the brain obtain this oxygen? How does the oxygen get to all the regions in the brain? Well, the brain has a very sophisticated system of blood vessels, like your whole body, of vasculature, that serves as an energy shuttle, okay? And these blood vessels are little tubes that are carrying the blood through all the regions in the brain and are delivering that oxygen to the cells so they can function. So you have arteries that are carrying the oxygen-rich blood to the body, to the brain, and you have veins that are then carrying the oxygen-depleted blood back to the heart so it can get reoxygenated, okay? So let's try and put those two things together. So I told you that the neurons communicate with electrical signals, and I also told you that they need oxygen, and the oxygen comes in the blood. So imagine that you have these neurons communicating with each other, and they're taking up the oxygen from the blood. They use up the oxygen, and then they tell the blood vessels, hey, I need more oxygen. So they send more blood there, right? And so now they can use up the oxygen again, and then we have the veins carrying the blood back that was depleted of oxygen so it can get reoxygenated. Okay, so that is happening when we have brain activity. The neurons fire, they use up the resources, and then those resources are replenished. And that is what we can detect with functional magnetic resonance imaging, so fMRI. And the signal that we detect, we're not looking at the electrical activity. We're looking at what regions in the brain are replenishing oxygen, which is associated to the activity. It's called a blood oxygen level dependent signal, a BOLD signal, okay? So the tool we use to do this is called an MRI scanner. Who here knows what an MRI scanner is? Who here has had an MRI scan? Right. So as you might know, an MRI scanner, typically you're looking at the structure of something, the structure of your brain, the structure of some other organ in your body. But this is a special type of MRI scanner that can look at this BOLD signal, right? This blood oxygen level dependent signal. So the question then becomes, how do we collect this data? How does it work? Okay? So the basic unit at which we collect the data is called a voxel, which is a three-dimensional cube. So just imagine a 3D cube. And we split your brain into a bunch of 3D cubes, okay? And we are collecting the signal at each of these little cubes. And so think about it as being analogous to a pixel. And a pixel is what makes up images. So this slide right here that I'm displaying is made up of a bunch of tiny pixels. Some of them, most of them are white. Some of them are black in the word voxel. Some of them are red in the red square there, right? So it's like a pixel in that way. So the MRI scanner is collecting the data in slices. It goes through slices of the brain. And if you put a bunch of voxels together, it will look something like that. If you put a bunch of pixels together, you can get an image, a 2D image of Mario like that. And so the scanner will collect all these slices, and eventually it'll collect all the slices. You put those together, it'll look something like that. Just like if you put a bunch of slices of Mario together, it'll look something like that. And then every 2 seconds, we collect data from the whole brain. And we go back and collect it again. And that is called a volume of data. And it might be analogous, right, to this nice 3D smooth Mario. Okay, so what can we do then? Well, like I said, we collect data from the whole brain every 2 seconds. So 2 seconds, 4 seconds, 6 seconds, and maybe we have 10 minutes of data. What I can do now is I can extract my signal, my BOLD signal, from, say, that voxel right there, okay? And I can plot it out, and I can see what it looks like. So I can do that, and we call that a time series or a time course, okay, of signal or activity. And so I can see how the activity in that voxel is changing over time. And we can do a bunch of cool things with that. One of the cool things that we can do is we can have you do a task inside the scanner. So we have you lie in the scanner, and we have a computer that is displaying some sort of task. And maybe the task here is I'm showing you a picture of fire. Maybe that's a task. And then we put some mirrors in the scanner or a headset with mirrors, and you can see the screen when you're inside the scanner. And then we can even give you maybe a button box so that you can respond to the task if we need you to respond. And now we can look at what regions in the brain are active during the task. So this is an example of this task-based fMRI where we're showing people either pictures of faces or places or scrambled pictures. And the scrambled pictures, what we're doing is we're moving around the pixels. See how I work pixels back into the talk? And so basically it's a scrambled image. So what we can do now is we can look at what brain regions are active when people are looking at faces. And in that way, we can study the brain regions involved in facial processing. And we can do this for a whole bunch of things like memory tasks, attention tasks, etc. Another cool thing that we can do, and this is really what I do, is we can have you do nothing in the scanner. Just lie in the scanner for 10 minutes, and we're going to see how the signal changes while you're at rest. And we call this resting state fMRI. You can see that scientists are really not that creative with the names. It's pretty straightforward with the naming. I'll take questions in just a little bit. And so resting state fMRI, what we can do, we can look at a brain region, say we're looking at that blue region there, and we can extract the signal from that region. Maybe it looks like that, right? And now I can compare it to the signal in other regions. So maybe I extract my signal from this other region, and it looks like that. Now, can anybody tell me, do these two signals look like they're going up and down together? Yes or no? Let's raise hands. So who thinks they're going up and down together? Raise your hand. Who thinks they're not going up and down together? Okay, so they are going up and down together. It's not perfect. It's not a perfect correlation, but it's pretty close. When two regions seem like they're fluctuating together, we say that they are functionally connected, that they show functional connectivity. And by doing this approach, we can study how brain regions are communicating with each other. Scientists think that when the signal goes up and down together in two regions, that they might be involved in mediating the same process in the brain, right? Or involved in the same sort of thing. A good analogy that we use to explain that is the speaker and receiver analogy. So I'm talking right now, so I'm emitting some sound waves, right? And you're picking those up with your ear, so you're the receiver. And my voice is fluctuating in a certain frequency, and then your ears are then vibrating at a certain frequency. And because they're going more or less in the same frequency, we can communicate. So that's essentially how we explain this, or how we think this is working, right, with the brain regions. So just a quick summary. I told you that the brain is responsible for directing behavior. I told you that the brain is made up of cells called neurons, that these neurons need nutrients and oxygen to function, that when they activate, they're going to use up the oxygen, and lastly, that functional MRI can detect what regions are replenishing that oxygen supply. And that's how we can look at brain activity, or at least a proxy of brain activity, because it's not directly the electrical signals. And so with that, I will take any questions. So I know we had a question over here at sort of the middle of the talk. Okay, never mind. So question over here.
What happens when you have more than one speaker? Would that make it even better? Would that make it even harder to hear the first person speaking?
What happens when you have more than one speaker? So I'm going to take that in the context of the brain and the communication between the brain regions. So yes, definitely we look at, when we do this type of work, when we're looking at the functional connectivity of brain regions, we can look at more than two regions at a time. We can look at, say, 300 regions. And we can look at how each of those regions seems to correlate with each other. And we can then look at large-scale patterns of connectivity in the brain. And we can look at brain networks, which are large-scale, sort of multiple regions, and how they communicate with each other. And it can get very, very complicated. It comes out to very large data sets and very large data. Another question over here.
What made you choose to study in Puerto Rico?
So I am Puerto Rican, born and raised. That's why I studied in Puerto Rico. I studied at the public university in Puerto Rico, so that was really important to me. A lot of hurricanes, a lot of stuff, but I stayed there and I did my bachelor's degree there.
How long does it take for the MRI to get one complete scan of the brain?
So every 2 seconds we get the whole data. But for one of these, for example, one of these resting scans, the conventional thought right now is that you need about 8 to 10 minutes of data in order to have the full or usable set of data, right? That's more or less what we think about. But some tasks take longer. So I know some tasks maybe last 8 minutes, maybe 25 minutes. Some people have folks look at movies inside the scanner. And maybe the movie lasts, 30 minutes, and I get 30 minutes of data. And the reason that we use movies is because some people argue that if I just tell you to line the scanner for 10 minutes, everybody's going to do something different. Maybe you're going to think about what you're going to have for lunch later, and then somebody else is going to be thinking about what they did in the morning or what they have to do in the afternoon. So a way that we can kind of get everybody on the same track is that we just have you look at a movie, and so everybody looks at the movie, so everybody's going through the same scenes and the same experiences, and in that way we can standardize a little bit that signal, and we can then compare between people or groups better. That's one way that we can do it.
Why does your brain need oxygen?
Why? That's a great question. So why does your brain need oxygen? I would think it just boils down to the cells in your brain, the neurons. These cells need oxygen to function, to produce energy. And because that's the fundamental unit at which the brain is sort of organized, then the brain itself, right, needs this oxygen so that the cells can function properly. And if you don't have the oxygen, the cells can't function, and so your brain can't function. That's a great question. Over here?
Does your blood cell or your blood oxygen level increase in the brain when you're thinking more? Or if you have a lot of activities and multitasking, does the oxygen level increase in your brain?
Right. So whenever you have some activity in the brain, that region is going to use up those resources, and then subsequently there's going to be an increase in oxygenation. And the key thing here that I did not mention is that the brain overcompensates. So it sends more blood there than it was used, right? And that is important because that is why we can do this technique, because of that overcompensation. It sends much more blood to that region than was used, just to make sure, that the resources are replenished, and then we can detect those replenishes of oxygen with the fMRI. Question, right?
If you have more brain activity, how does it communicate faster with the neurons in the brain?
If you have more brain activity, does it communicate faster? So this is all happening pretty fast. The timescales that we're talking about here are really, really, really quick. So that is one of the limitations of this technique. So like I told you, I can only look at changes that are happening at the level of 2 seconds, right? That is not the level at which the brain is functioning or the neurons are functioning. Things are happening much faster than that. But the best we can do with this technique is look at changes every 2 seconds. So that is a limitation here. But the brain is much, much faster than that. Yep. Good question. Here.
Can you comment quickly on your field of study with fMRI? fMRI.
What I study? Okay. So I study fMRI and I use it to study substance abuse disorders. So I mostly do resting state fMRI. So we look at, right, at rest, how the activity is fluctuating. And what I've done in the past as a post-bac fellow is that I've looked at the patterns of connectivity. So if I, instead of having just 2 regions, say I have 200 regions, and then I relate every single region to every other region, and I can see how correlated their activity is, and I do that for all 200, and then I can look at what are the recurring patterns throughout the 10 minutes of connectivity, right? and I can see if I find any relationships between those patterns of connectivity and some behavioral or affective readout, right? So maybe how the person is feeling in terms of their anxiety or the depression, or maybe how they're performing in a memory task. And I try to see if the patterns are predictive of any of that. I don't know where, so over here?
How does the brain know that we're inhaling oxygen?
How does the brain know that we're inhaling oxygen? So there are oxygen sensors, and I might be wrong about this, but it's my best. There are oxygen sensors in the brainstem, if I'm correct. And so, say you hold your breath for a bit. Eventually, your body's gonna tell you, okay, breathe. And that's because there are sensors that are in the brainstem that can detect that sort of decrease in oxygen, and they'll then tell the brain, hey, you gotta breathe, and then you breathe. And so, yeah, the brain can detect that.
Yeah, I got a question, please. So I saw you used rats for your experiment. Can you hear me?
Yep, I just don't know where it's from. Okay.
Yeah. So you use rats for the experiment.
Mm-hmm.
So what other animals do you think are more effective? Do you use birds also?
So neuroscientists use a lot of animal models. A lot of the work is done in mice and rats and sort of rodents. Some work is done in birds, definitely. Some people use primates. It really depends on the question that you want to answer and whether or not that animal model is helpful to answer that question. So, for example, a lot of research is done in flies, and that's just because flies are very easy to manipulate genetically. We can manipulate flies' genes very easily. So it kind of depends, right? So when you're working in primates, you're typically, because you want to look at some behavior, and primates are really close to humans, so primates can learn a lot of different tasks that other animals cannot learn. Very complicated tasks, actually. But yeah, great question.
Okay, so we have one more question up here, and then we'll have to move on.
Right.
Does drugs and alcohol affect the oxygen to your brain? Like, how bad does it?
Right. So that is a really good question. So that is a potential confound when we do this research, right? In a lot of different conditions, say people are smokers or they drink alcohol or some other condition, the question when you're doing this type of research, the first question is, do I expect some sort of change in oxygenation? And that would then affect my signal, and that would not be related to brain activity. It's just purely an effect of differences in oxygenation. And so that is a very important question that people look into consistently, and they have to make sure, if I'm going to study something with this technique, I have to be sure that my two groups are not different in terms of oxygenation, because that's going to confound my signal. So that is certainly a question that scientists struggle with. All right.
Thank you so much,
thanks,
Lester,
everybody.
for presenting this amazing talk.